So let's carry on. Right. So you've got to read this passage. At the beginning of the 20th century, maggots were widely used to treat wounds. Then penicillin started in the 1940s. And then you read that, and you read that. Typical wound dressing. Then they ask you what is a pathogen. No. So it's antibiotic, not pathogen. Antibiotic. What is an antibiotic? A substance that kills pathogens. Well, let's use the word bacteria. A substance that kills bacteria. Name 2 antibiotics. Ooh, right. Penicillin. And there was another one. Mirabilicide. Okay. Penicillin and mirabilicide. And methicillin. No. Yep, 3. Any one of those. Right. You can write any one of those. We'll write penicillin and methicillin. I think that's how you spell it. So this is why maggot are bred in a sterile environment. This is to stop them being infected with pathogens and passing it on to the patient and passing it on to the patient. Why are green blowfly maggots chosen to treat wounds? Because they only eat dead meat, dead flesh. So they won't, if the flesh that's dead, it'll clean that up and it won't touch life, so it won't burrow into the flesh. Right. Bacteria are made out of proteins. So the enzyme for proteins will be what? We can say protease. That's the group of enzymes. If you put in pepsin or trypsin, the 2 that I mentioned in the book, you'll be alright. Right. Describe how the body's immune system kills the bacteria. There's 4. So remember, first thing that happens, the lymphocytes, what do? They produce what? Produce antibodies for specific antigens. Then some lymphocytes become what? Become memory cells and stay in blood, providing immunity from future infection. Then the phagocytes, what? They engulf. So remember these words: engulf and digest the pathogens. Right, there you go. Let's see if that's enough points. Yeah, what would be good here right at the beginning if we said the white blood cells. Right, the white blood cells, and then which one? Lymphocyte, and then which one? Phagocyte. Right, Give a disadvantage of using Magabax. So it takes too long. It takes long and is expensive. Right. So now here you've got this question, and a lot of it's just a different version of it. A lot of this comes up. So what you've got is use your knowledge of natural selection to explain why you've got superbugs. These are called superbugs. So this is how you answer this. You say it's just the standard structure. Yeah? So random mutation in DNA leads to variation in the superbug in MRSA bacteria. This mutation, let's use the word variation. This variation means what? This variation means the bug or bacteria, let's use the word bacteria, survives the antibiotic treatment, the antibiotic. Right. If it survives, it then breeds, passing on its resistant allele or gene. Okay, gene, you can put gene in there. Passing on its resistant allele to its offspring. So it's very particular, this structure. This continues on for generations. This continues on for generations. That's how you get lots of superbugs. So just remember the structure. Watch this. Look. Random mutation in the DNA leads to variation. This variation means that the bacteria survives because it becomes resistant to it. Yeah. Right. Because it survives, it then breeds, passing on its resistant allele. It passes. What's going on? Resistant allele to its offspring, and then this continues on for generations, and then what happens is you get lots of superbugs, then lots of them, not just one. Oh dear. Right, let's try this. The body uses white blood cells to combat infection. One type of blood ingests. Oh, not very good, is it? Hello. Oh no. Raboya. Right, let's just stick to a dark. No, that would be a good color. Modify tool. Right, it ingests it. Which ones ingest it are called phagocytes. After ingesting it, they produce a digestive enzyme that digests the bacterium, breaks them down, while the other white blood cells are called lymphocytes. And they release specific molecules that bind to antigens on the surface of the pathogen. Other components of the blood are also involved in helping wounds to heal. Small cell fragments are called platelets to help the blood clot. This seals the wound to prevent further bleeding and prevent the entry of pathogens. This is a good one, actually. If you can go over this, it covers everything really nicely, actually. That's good. Right, how many have we got? 7, 8, 9. Oh my god. Who put this I put this together. Silly me. Right, come on. I feel like one of my students now. Looking for an excuse. Right. Fish farms remove nitrogenous waste to improve the growth of fish. Another method to improve is vaccination. Explain how the process of vaccination improves. Right. You inject or okay, let's use the word. You inject an inactive pathogen. Well, because it's a fish, you wouldn't inject it, you'd feed it in its food. Inactive pathogen. The lymphocytes produce antibodies and some become memory cells, and this is the key part to memory cells. These memory cells stay in the fish, and if the pathogen infects the fish again, the memory cells produce lots of antibodies and in larger numbers to fight off the pathogen. This is called the secondary immune response. And that should do it. So what happens? You feed or why does you know what's happened? Something's happened, it doesn't like it. Oh, it does now. You inject an inactive pathogen, not a live one, because that might cause disease and cause it to spread. The lymphocytes, then the white blood cells, the lymphocytes produce antibodies, and also some become memory cells. It's these memory cells, I'm supposed to put cells there, stay in the fish, and if the pathogen ever infects the fish again, the memory cells produce lots of antibodies and no, come on, you can't use that twice. And quickly produces it quickly. This is called the secondary immune response. All these keywords you got to know. Put them into your head. And let's move on. So that's 13. Okay, I think we're going to have to. Let's try that. Right, let's get this one done and then we can set another video up. The structure of the bacterium differs from the structure of a virus. Two structures found in a bacteria, not found in a virus. So you've got a cell wall and flagellum. So you've got the cell wall here, and you've got the buccal, the tail, flagellum. Right. So some pathogenic bacteria. So that's your bacteria. This here is a white blood cell. Now they've not named it. So what you do is you say the phagocyte engulfs the pathogen. It then releases what? Digestive enzymes and kills the pathogen. So it's a 3 marker. Right, okay, that's it, that's enough. So the key thing here is phagocyte engulfs, releases digestive enzymes, and then kills the pathogen. There's other things you could put here as well. You could put in cell membrane, cytoplasm, you've got plasmid and other things, but those are the 2 most obvious ones. So you've got a plasmid here, you've got some more DNA material here, you've got nucleoid, you've got cytoplasm in here inside, you've got inside you've got like a plastic bag called the cell membrane in there. Right, we're going to call it quits there.